Welcome to the deep dive. This is your shortcut to getting, well, properly informed. Today, we're diving into a world that's usually unseen, microscopic even, but its impact. It shapes so much of our daily lives. Think about the bread you eat, maybe the wine you drink, even life-saving medicines. They're often connected to this hidden world. So our deep dive today is into Hemiascomycetes. They're a really key class of fungi, mostly yeasts. And we're using a chapter from Introduction to Fungi, the third edition, as our guide. It's, pretty dense scientific stuff. Our mission, though, is to unpack it all for you in a way that's clear, hopefully engaging. We want to bring these tiny processes to life, just using words. Especially helpful, we hope, if you're maybe a college student tackling these very concepts right now. We'll flag the real world importance too.
Exactly. And for this deep dive, we'll sort of build up the understanding step by step. We'll look at their structure, their morphology, what makes them unique, then their life cycles, which can be quite diverse, plus where they live, their ecology, and crucially, their massive contributions. Medicine, industry, they're surprisingly important. And yeah, we'll weave in the key terms as we go so it feels natural, not like a vocabulary list.
Okay, let's get into it then. Hemiascomycetes. What really makes them stand out from other fungi, like the ones that make mushrooms we see?
Right. Well, the classic examples are the yeasts within the Ascomycetes group. And a really key difference compared to what we sometimes call higher Ascomycetes or Euascomycetes, it's about structure, or rather what they lack. Think about a mushroom or maybe a morel, those complex fruit bodies. Ascomycetes make those. They have intricate structures, these things called ascogenous hyphae, and the spores are enclosed in a protective body, an ascocarp. Hemiascomycetes, though, they're much simpler. They're ascomycete sacs holding the spores. They basically form freely, singly, almost naked, you could say. No elaborate housing.
Ah, okay. So a big structural difference right off the bat. What about like their cell walls or inside the cells? Anything distinct there?
Yeah, that's fascinating too. Their cell walls have surprisingly little chitin.
Really? I thought chitin was big for fungi.
It often is, but here sometimes it's just a small ring left behind where a new daughter cell buds off. That's quite different. And if you could look even closer inside at the connections between cells, if they even form hyphae, which many don't. The pores, the septal pores, they're usually tiny or plugged, and they're missing these specialized stoppers called Woronin bodies.
Woronin bodies.
Yeah, think of them as safety plugs in other fungi. Their absence here means there's basically no cytoplasm flowing between adjacent cells in a hypha, which is different from Euascomycetes, where even organelles, sometimes nuclei, can pass through those connections.
Okay, that raises a practical question then. If they don't have these big, obvious structures and internal communication is limited, how do scientists actually identify them? Especially, you mentioned if they don't always make spores easily in the lab.
That's a great point.
Yeah.
It absolutely can be tricky. Without seeing clear asci and ascospores under the microscope, identification is tough. So, historically, researchers used a lot of physiological tests, like seeing what sugars or nitrogen sources a particular yeast could metabolize. It was kind of painstaking.
I bet.
But no. Routine DNA sequence analysis is really common, especially looking at the 18S ribosomal DNA gene. It's become standard practice. It allows for really precise identification, often right down to the genus, and it clearly separates them from other fungal groups. No ambiguity.
Right, the power of genetics. So these often naked ascus yeasts,
Yeah.
where do they actually live? Are they just lab curiosities or are they out there in the world?
Oh, they're absolutely out there. They're truly cosmopolitan. Ubiquitous is the word. You find them almost everywhere. They're really prominent on plants, particularly as what we call epiphytic saprotrophs, meaning they live on the surface, feeding on dead organic matter or secretions.
Epiphytic on the surface?
Exactly, especially where there are sugars readily available. Think flower nectar, ripe fruits, even just sap oozing from a wounded tree branch. The numbers can be staggering. You can find hundreds of thousands, sometimes millions, of yeast cells per gram of plant material.
Wow. Okay, so plants are a major habitat, but you said ubiquitous. Where else do they show up? This is where it gets really interesting, I think.
Yeah, beyond plants, they're definitely in the soil, often getting washed in or mixed in with decaying plant stuff. They're found in fresh water. They're found in marine environments. Lots of species are associated with insects, too, and other animals, including the guts of vertebrates. We have a whole community of yeasts living inside us.
Including us humans?
Yep, some even live right on our skin surfaces, part of our normal microflora.
And that brings us naturally to one species many people might have heard of, maybe not in the best context. Candida albicans.
Exactly. Candida albicans is one we definitely need to talk about. It's usually a commensal, right? Lives on us harmlessly most of the time.
Usually, yeah.
But under certain conditions, especially if someone's immune system is weakened, it can switch roles and become a pathogen, causing anything from mild issues to quite severe infections. Thankfully, most Hemiascomycetes aren't major plant pathogens. There are a few exceptions, like some Aromethecium species that cause spots or lesions on crops, like citrus or cotton, but it's not their main ecological role.
Okay. And what about food? I've definitely had, fruit go weirdly fizzy. Is that yeasts? And is it a big problem?
It very likely is yeasts. Many Hemiascomycetes are pretty good at growing even when water is scarce. Think really sugary jams or salty brines. Osmotolerant, we call them.
Osmotolerant, right.
So yeah, that allows them to colonize lots of preserved foods. The good news, generally, is that yeast spoilage doesn't usually involve toxin production, unlike some molds or bacteria that can produce really nasty stuff. So it's more of an economic loss, a quality issue.
Still significant, though, I imagine.
Oh, definitely. The economic losses from yeast spoilage worldwide are considerable. It's a real issue for the food industry.
And for, say, students wanting to study them in a biology lab, are they easy to get hold of and grow?
Yeah, relatively speaking, they are. They grow readily on standard lab agar media. You usually add some antibiotics to keep the bacteria down. You can take a sample from fruit skin, soil, whatever, plate it directly, or maybe suspend it in some water first. And importantly, they're just big enough to actually see as individual cells with a basic light microscope, maybe at 100x or 400x magnification. You can distinguish them from tiny bacterial cells. That's true.
Got it. So they're everywhere, doing all sorts of things. But let's pivot to the impact on us. Why are these tiny Hemiascomycetes, especially maybe just a handful of species, so incredibly important in biotechnology in our daily lives?
Right, this is where their positive impact really shines. And we have to start with probably their oldest and still maybe most important contribution, alcoholic fermentation.
Yes, beer and wine.
Beer and wine, primarily driven by Saccharomyces cerevisiae, billions upon billions of liters produced globally every year. And what's amazing is humans seem to have stumbled upon this process using yeasts multiple times independently throughout history all over the world.
It's incredible. I remember reading theories that the desire for beer might have even been a driving force for early agriculture, like thousands of years ago.
That's absolutely a serious theory some historians propose. The stability in calories from grain turned into something enjoyable and safer than water sometimes. It might have been a huge incentive. And the process continues today, refined, of course. Different strains of Saccharomyces for top fermenting ales versus bottom fermenting lagers, each giving unique flavors. Even things like the old German purity law for beer, the Reinheitsgebot, though it's formally gone, it just highlights how deeply embedded this yeast is in culture and economics.
And it's not just about the alcohol, is it? They do other fundamental things for our food.
Exactly. Their role in bread making is just as vital. It's not the alcohol we're after there, it's the carbon dioxide they produce during fermentation. That's what makes bread rise and gives it that light, airy texture.
Right, the leavening.
Precisely. And the scale is immense. Something like 1.5 million tons of fresh Saccharomyces cerevisiae baker's yeast produced globally each year just for dough.
Wow, 1.5 million tons of yeast.
Just for bread. Then there's another angle, single-cell protein, or SCP. This involves growing yeasts, often on industrial byproducts or waste streams, things like whey or molasses or paper pulp waste. They convert this low-cost stuff into a biomass that's really rich in protein and vitamins. It can be used as animal feed or sometimes even processed for human food supplements.
And they're safe for that.
Generally, yes. A big advantage is that yeasts like Saccharomyces or Pigia jejuni don't typically produce mycotoxins, unlike some filamentous fungi, which makes them much safer as a food source.
Okay, so fermentation, bread, protein. What else are these microscopic powerhouses up to? Any medical applications?
Oh, absolutely. Some are used directly for vitamin production, like certain Aromethecium species are used industrially to produce riboflavin, which is vitamin B2. But maybe even more significant today is their use in producing recombinant proteins. This is huge for modern medicine.
Recombinant proteins, like what?
Things like therapeutic enzymes, antigens for vaccines, hormones like insulin, even growth factors like epidermal growth factor. Instead of extracting tiny amounts from animal tissues, we can insert the human gene for that protein into yeast cells, and they become little factories, churning it out.
Wow.
And a particular yeast, Pichia pastoris, is really valuable here. One reason is that the way it adds sugar chains glycosylation to the proteins it makes is very similar to how human cells do it. This is important because it means the final protein product is less likely to cause an immune reaction when used clinically in humans.
That's incredibly clever. Any other surprising jobs for these yeasts?
One more area is biological control, biocontrol.
Using them to fight other microbes.
Exactly. Remember how they colonize plant surfaces like fruit skin? And they generally don't produce toxins. Well, researchers are developing certain yeast strains, like Pichia guirmandii, to spray onto harvested fruits. The idea is that the yeast harmlessly takes up space and nutrients, potentially outcompeting or even inhibiting the growth of molds that cause spoilage during storage and transport. A natural fungicide, essentially.
Okay, let's zoom back in on the star player, Saccharomyces cerevisiae. Baker's yeast, brewer's yeast. It's obviously huge in biotech, but you also mentioned it's a major scientific model organism. Why is it so important for just basic biology research?
Well, a huge milestone was that S. cerevisiae was actually the very first eukaryote, an organism with complex cells like ours, to have its entire genome sequenced. That was back in 1996.
The first eukaryote, wow.
Yes. That complete genetic blueprint, combined with the fact that it's relatively easy to grow and manipulate genetically, in the lab you can knock out genes, add genes, modify them, has made it an incredibly powerful tool. It's become a fundamental workhorse for understanding all sorts of basic eukaryotic cell processes. How genes are regulated, how cells divide, how organelles function. A lot of what we know about our own cells we first learned by studying yeast.
So can you walk us through its life cycle? You mentioned budding, but also a sexual cycle. How does that work? It sounds more complex than just splitting in two.
It definitely is. So the cells you usually encounter doing fermentation or budding are generally diploid. They have two sets of chromosomes just like most of our body cells.
Okay, diploid.
And they reproduce asexually just by budding. A small outgrowth forms, gets bigger, pinches off as a daughter cell. Under good conditions, they can do this really fast, maybe doubling their population every 100 minutes or so.
Pretty quick.
But then there's a sexual cycle. This usually starts with haploid cells, called ascospores, which have only one set of chromosomes. These haploid cells confuse together. But here's the interesting part. They have mating types. Think of it like two sexes, though we call them A and alpha. It's controlled by a specific SCOT on their DNA, the mating type locus.
An alpha.
Right. And they can only fuse if they're opposite types. When an A cell comes near an alpha cell, they start communicating. They release little peptide hormones called pheromones. Each cell type responds to the pheromone of the opposite type. This causes them to stop their normal budding cycle and instead grow towards each other, forming sort of a projection.
They reach out to each other.
Exactly. They grow towards the source of the pheromone, eventually touch, their cell walls and membranes fuse, and their nuclei combine. Bingo, you've formed a new diploid cell, which is heterozygous at the mating type locus.
Okay, so they have asexual budding and this more complex sexual fusion. What's the point of the sexual cycle? How does it help them survive, especially in tough times?
That's the key question. Survival. It turns out that only those diploid cells that are the result of mating the alpha cells are capable of undergoing meiosis when conditions get harsh, like when nutrients run out.
Meiosis, that's the process our own cells use to make sperm and eggs, right? Having the chromosome number.
Precisely. In yeast, meiosis leads to the formation of typically 4 haploid ascospores, usually packaged together inside the remnants of the original diploid cell wall, which now acts as an escus. And these ascospores, they are much, much tougher than the regular vegetative cells. They're resistant to heat, drying, starvation. It's their survival capsule, allowing the species to wait out bad times.
Okay. So sex leads to survival spores.
Yeah.
Now, I remember hearing something really wild about yeast, that they can sometimes change their mating type. Is that real, and how does that fit in?
Yes, it's one of the most fascinating things about S. cerevisiae.
No.
Many strains possess this incredible ability for a single haploid cell to switch its mating type.
How does that even work?
Imagine you start with a single haploid cell, say type A. It buds off a daughter cell, which is also type AA. But the mother cell, before it divides again, can switch its genetic information at the mating type locus, changing itself into an alpha type.
The mother cell changes.
Yes. So now, in the population growing from that single original cell, you have a mix of A and alpha cells present. This dramatically increases the chance that cells will find a compatible partner nearby, fuse, form those diploid ancels, and then be ready to make those tough ascospores if needed.
That's amazing genetic flexibility.
It really is. It involves a complex mechanism often called the cassette model. The yeast cell actually carries silent, hidden copies of both the A and alpha genes elsewhere in its genome. And there's a specific enzyme system that can basically cut out the gene currently active at the mating type locus and swap in a copy of the other type from the hidden cassette. It's a highly regulated gene switching system.
Wow. Okay, let's switch from genetics to structure again. We can't see diagrams, but can you describe the cell wall and the budding process in a bit more detail? You said it's complex.
Sure. Think of the S. cerevisiae cell wall as being quite substantial, maybe 15–25% of the cell's dry weight.
Yeah.
It's multi-layered. The main structural backbone is made of branched glucan polymers, complex sugars. Interwoven with this is some protein and, as we mentioned, very little chitin, mostly concentrated just in the neck region where the bud forms.
Glucans and proteins mainly.
Right. And the outermost layer is often rich in heavily glycosylated proteins called manoproteins. These outer proteins are important. They determine things like how sticky the cell surface is, its overall charge, and what can pass through into the cell.
And the budding itself, how does that intricate process unfold?
It's really dynamic. It starts at a specific site. First, a sort of cap of proteins assembles. Then, around this cap, a ring of proteins called septins forms. This septin ring stays at the neck between the mother and the growing bud.
Septins. Like a collar.
Exactly, like a collar or a scaffold. As the bud starts to swell outwards, other proteins direct the growth. Crucially, inside the cell, there are these filaments called actin cables. Think of them as miniature conveyor belts. They actively transport vesicles, little membrane sacs filled with new cell wall material and enzymes, specifically to the tip of the growing bud. That's how it expands.
So targeted delivery of building blocks.
Precisely. Meanwhile, the nucleus has to divide. This is guided by other internal structures called microtubules. One copy of the nucleus moves into the bud. Finally, that septin ring at the neck helps to constrict and eventually pinch off the connection, separating the daughter cell. This process leaves a permanent mark on the mother cell, a bud scar.
A scar. Like a belly button.
Sort of. And a mother cell can only accumulate a certain number of these scars, maybe 20 or 30, before it stops budding and eventually dies. It suggests that even single-celled yeast have a finite reproductive lifespan. They age.
Fascinating. So that's budding. But you also mentioned they can form pseudo-hyphae. What's that about, and why do they do it?
Right. Under certain conditions, often when nutrients like nitrogen are limited, S. cerevisiae can switch its growth mode. Instead of budding off individual roundish cells that separate easily and form a smooth colony on an agar plate, the budding process changes. The daughter cells become elongated, and they don't fully separate from the mother cell. They remain attached, and then they start budding in a similar elongated way.
So they form chains.
Exactly. Chains of elongated connected cells. That's a pseudohypha. And importantly, these chains can grow invasively. They can actually penetrate down into the agar surface or whatever substrate they're on.
Why would they do that?
It's thought to be a foraging strategy. When nutrients are scarce on the surface, growing downwards allows the yeast to explore a larger volume, potentially finding more food deeper in the substrate. It's an adaptation for nutrient acquisition.
Clever. Okay, one last deep dive inside the cell. You mentioned internal membranes and the vacuole. Can you tell us more about how yeast manage all that internal traffic and waste?
Yeah, there's been a huge amount of research on yeast membrane trafficking, partly because it's so similar to our own cells. It's like a highly organized intracellular postal system. Proteins, destined for secretion or for insertion into membranes, start their journey in the endoplasmic reticulum, then move through the Golgi apparatus for processing and sorting. From the Golgi, they're packaged into tiny membrane vesicles that bud off and travel to their specific destinations. Maybe the cell surface to be secreted, or maybe to the vacuole.
And stuff comes in too, right? Endocytosis.
Absolutely. The cell surface can invaginate, forming little pits that pinch off inwards, bringing in molecules from the outside, or recycling bits of the cell membrane itself. This process, endocytosis, is crucial for things like taking up nutrients, or responding to signals like those mating pheromones we talked about. And much of this traffic, both coming in endocytic and going out secretory, converges on the vacuole.
The vacuole, I usually think of that as just a big storage sac in plant cells. Is it more complex in yeast?
Oh, much more. In yeast, the vacuole is a really dynamic, multifunctional organelle. It's definitely a major destination for membrane traffic, receiving vesicles from both the Golgi and the endocytic pathway. Think of it as the cell's main recycling center and waste dump. It's loaded with powerful digestive enzymes, hydrolases that can break down proteins and other macromolecules.
So it digests stuff.
Yes. Material delivered there gets degraded. This is vital for recycling nutrients, especially during starvation. The vacuole can even engulf and digest parts of the cell's own cytoplasm in a process called autophagy self-eating to survive extreme conditions. But it's also a storage depot. It can accumulate reserves of amino acids, phosphate, and other essential building blocks. It plays a role in detoxification, sequestering potentially harmful ions like heavy metals, keeping the cytoplasm safe. Its shape and size can change dramatically depending on the cell's needs and environment. It's really a central hub for cellular housekeeping and adaptation.
Incredible. So, Saccharomyces cerevisiae is clearly a model organism and a biotech workhorse. But it's not the only Hemiascomycetes out there. Are there other members of this group with equally fascinating stories or impacts?
Definitely. The diversity is huge. Let's touch on killer yeasts, for instance.
Killer yeasts. Sounds dramatic.
It kind of is. These are strains, often of Saccharomyces, but also other genera, that produce toxins that kill sensitive strains of yeast, even closely related ones.
They kill their competition.
Essentially, yes. And the really neat part is the killer strain itself is immune to its own toxin.
How does that work? In Saccharomyces, you mentioned viruses.
In some common Saccharomyces killer strains, yes. The toxin is actually encoded by a genetic element that's essentially a double-stranded RNA virus living permanently inside the yeast cell. This virus provides the blueprint for the toxin protein and also for the immunity factor.
And the toxin itself, how does it kill another yeast cell?
Let's take the well-studied K1 toxin. It's made as a precursor protein inside the killer cell, gets processed and modified as it moves through the secretory pathway, and then gets secreted outside. Once outside, it finds a sensitive yeast cell. One part of the toxin, the beta subunit, latches onto a specific component of the target cell's wall, a type of glucan. Then the other part, the alpha subunit, inserts itself into the target cell's plasma membrane and forms an ion channel, a pore. This disrupts the cell's crucial ion gradients, particularly proton flow, and ultimately leads to cell death.
And the killer cell avoids this.
Right. The producing cell has its own toxin receptor, but it's modified or somehow masked, preventing the toxin from forming lethal pores in its own membrane. It's a sophisticated system.
So what's the point in the real world? Is this just microbial warfare?
Pretty much. It gives the killer strain a significant competitive advantage, especially in environments like sugary fruit surfaces, where lots of different yeasts are competing for limited resources.
Any practical implications?
Well, in industrial settings like winemaking, killer strains can sometimes be a problem if they show up unexpectedly and kill off the desired fermenting strain. But there's also interest in potentially using these toxins or synthetic versions or antibodies that mimic them, perhaps as antifungal agents, maybe against problematic yeasts like Candida. Research is ongoing there.
Speaking of Candida again, Candida albicans, you mentioned it's a commensal but can become a pathogen. What makes it so adaptable, so good at living in different parts of the human body?
A key feature of Candida albicans is its dimorphism, or maybe more accurately, polymorphism. It can switch between different growth forms.
Dimorphism, two forms.
Yeah, classically it switches between a single-celled yeast form and a filamentous form, true hyphae with septa. It can also form pseudo-hyphae, which are sort of intermediate. And this ability to change shape is really critical to its ability to cause disease, its pathogenicity.
How so?
Generally, the yeast form is thought to be better for dispersal, spreading through the bloodstream, for instance. But the hyphal form is considered crucial for invading tissues. Hyphae can physically penetrate into host cell layers.
So, shape-shifting for attack.
Exactly. And the switch is reversible. It's triggered by environmental cues. Things like body temperature, 37 degrees still, tend to promote hyphal growth, while lower temperatures favor the yeast form. pH, carbon dioxide levels, nutrient availability, they all play a role. Conditions that mimic being inside the host, like in the bloodstream, push it towards hyphae. Conditions more like the skin might favor yeast growth. Plus, the hyphae express specific proteins on their surface called adhesins that help them stick tightly to host cells. And they secrete enzymes like proteases and phospholipases that can break down host tissues, aiding invasion.
And what about its sex life? You mentioned it was a mystery, but they found the genes.
That's right. For the longest time, maybe over 100 years, Candida albicans was considered strictly asexual, an imperfect fungus, just reproducing by budding. But then when its genome was sequenced, surprise, it had a full set of genes that looked very much like the mating type locus and pheromone response pathway genes found in Saccharomyces.
So it can have sex.
Well, it's complicated. It seems to be naturally diploid and doesn't readily undergo meiosis to form spores like Saccharomyces. A conventional sexual cycle hasn't been easily observed, but evidence strongly suggests a kind of parasexual cycle might occur. This could involve fusion of diploid cells, followed by gradual random loss of chromosomes to eventually return to a diploid state. It's not fully understood, but mating definitely seems possible, even if cryptic. And adding another layer of complexity is something called phenotypic switching or colony switching.
Switching colonies.
Yeah, a single strain of Candida albicans can spontaneously switch back and forth between different colony appearances when grown on certain lab media. The most studied is the white opaque switch. White colonies are the standard form, but opaque colonies look different, and the cells within them have different properties. They're shaped differently, stick differently, and crucially, have very different interactions with the host immune system.
And this affects mating.
Dramatically. Opaque cells are something like 1,000,000 times more efficient at mating than white cells. So this switching ability, creating different pre-programmed cell types within a population, likely plays a huge role in Candida's adaptability and success as both a commensal and a pathogen.
Given it is a significant pathogen, how do we actually treat Candida infections? And what about drug resistance?
Treating fungal infections, including Candida, is inherently challenging because they're eukaryotes, like us.
Right, our cells are quite similar.
Exactly. Compared to bacteria, there are fewer unique targets in fungal cells that drugs can attack without also harming human cells. This limits the number of effective antifungal drugs, and some that we do have can have significant side effects. Many current drugs target the synthesis or function of ergosterol. That's a specific lipid found in fungal cell membranes, but not in animal cell membranes, making it a good target.
Argosterol. Okay.
The big problem now is resistance. Candida species, especially albicans and others like glabrata, have become adept at developing resistance mechanisms. For example, against azole drugs, which inhibit an enzyme needed to make ergosterol, the fungus might start overproducing that enzyme, or the enzyme itself might mutate so the drug doesn't bind well anymore. Another major mechanism is actively pumping the drug back out of the cell using membrane proteins called ABC transporters. The cell just spits the drug out before it can do damage.
Like cellular bilge pumps.
Precisely. And resistance is a growing concern because the incidence of serious fungal infections has increased, especially in hospitals and among immunocompromised patients. But our arsenal of effective drugs is limited, and resistance is spreading.
A serious challenge. Okay, to wrap up our tour of this diverse group, are there just a couple of other Hemiascomycetes genera worth a quick mention?
Yes, definitely a few more highlights. The genus Pichia, we already mentioned Pichia pastoris for recombinant protein production. Another species, Pichia jardini, which used to be called Candida utilis, is important as a food yeast, used in making single-cell protein. Then there's Galactomyces candidus, often known by its old name, Geotrichum candidum. This one is very common, often looks mold-like. It's famous for causing sour rot in fruits and vegetables, breaking them down into this creamy, often sour-smelling mush. You see it on spoiled tomatoes sometimes.
Ah, sour rot. Not pleasant.
Not pleasant at all. Then, Saccharomycopsis fibrigera. This one's interesting because, unusually for yeast, it secretes enzymes called amylases that can break down starch, so it can grow well on things like flour or grain. It's also sometimes called a predaceous yeast because on loose surfaces, it can actually attack and digest other yeast species using cell wall degrading enzymes.
A yeast that eats other yeasts.
Yep. And finally, the genus Erymathecium, which we touched on briefly. These are notable mainly because they include the few significant plant pathogens in the Hemiascomycetes. They cause diseases like stigmatomycosis in cotton or hazelnuts, often spread by insects that puncture the plant tissues. But balancing that out, some Erymathecium species are also used industrially, as we mentioned, for producing vitamin B2, riboflavin.
Wow, what a journey through such a seemingly simple group of organisms, from naked asci and simple structures to complex life cycles, killer toxins, shape-shifting, and these huge roles in everything from ancient brewing to modern medicine and food production. Hemiascomycetes are clearly much more complex and impactful than you might guess from their tiny size.
Absolutely. And if you think about the bigger picture, it just highlights how incredibly interconnected our lives are with the microbial world. There's so much sophisticated biology happening at that microscopic level, and we're still learning so much from these organisms.
Yeah.
Whether it's harnessing them for biotechnology, figuring out how to combat the ones that cause disease, or just, appreciating the unseen ecological roles they play all around us.
So, reflecting on our deep dive today, what's the one thing that really stands out to you? Maybe the sheer scale of yeast used in bread, the cleverness of mating type switching, or perhaps the urgent challenge of antifungal resistance.
It is hard to pick one. But maybe it's that underlying theme of adaptability, the way Saccharomyces switches mating types, the way Candida switches form, the way killer yeasts compete. It makes you wonder, given this incredible adaptability and the diverse chemistry we've already harnessed from just this one group, What other microbial powerhouses are out there, maybe just waiting to be understood? And how might they completely reshape aspects of our future in medicine, industry, or ecology? That's the really provocative thought for me.
A great point to ponder. Thank you for joining us on this deep dive into the world of Hemiascomycetes. We hope you feel a bit more informed, maybe a lot more curious about fungi now.
And we look forward to exploring another topic with you in our next deep dive.